Assalamu alaikum. Have a good day to everyone. I welcome to all of you on 4th International Conference on Applied Zoology. It's Muhammad Amjad here, currently doing PhD in Zoology from Government College University, Faisalabad, and I have done this work at Institute of Pure and Applied Biology, Bahauddin Sky University, Multan. The topic of my presentation is the next generation sequencing identifies pathogenic variants in HGF, which is hepatic growth factor, POU3F4, which is neurotranscriptional factor, and TECTA, which is related with the tectorial membrane, and MY07A, which is related with the myosin proteins in consequence Pakistani deaf families. First of all, we will discuss about little bit the introduction. The congenital diseases. Congenital diseases, these diseases are basically attributable to the genetic causes. The incidence of the congenital diseases is known to be higher in families with contiguous marriages. The congenital diseases, there are a lot of numbers, but the final one and the more number, which are the congenital deafness. The congenital deafness, they are basically related with the consequence marriages, and these consequence marriages which we have investigated, they are related to the genetic causes, and these genetic causes reveal that there is prelingual severe profound deafness, and ultimately 1% to 2% of newborns throughout the world, and among them approximately 70% of the deafness is attributable to the genetic causes. The genetics of the hearing loss is extremely heterogeneous as to date. More than 120 genes are reported to cause non-syndromic hearing loss. It means that the hereditary hearing loss, which are similarly numerous genes which are known to be cause the syndromic hearing loss. In recent years, the next generation sequencing has been increasingly implemented in the genetic diagnosis of the heterogeneous diseases, and it reveals that there are pathogenic variants which including the hearing loss providing a high through output efficient approach, and to target the hundreds of the causative agents and genes which are related to the whole exome for the identification of pathogenic variants. In details, we will discuss about the material and methods. In material and methods, we have gone through the subjects and the clinical evaluations. In subjects, in material and methods, the first one is the subjects and the clinical evaluations. Three different families, which are consequence families, which are collected from Muzaffargarh, Rajanpur, and Okada, which are present in the province Punjab, Pakistan, with multiple individuals suffering from deafness. Written informed consent were obtained from all the participants and their parents before their enrollment in this study. Family members affected with the deafness were examined in the ear, nose, and throat, which are basically at the district headquarter hospitals by the medical specialist, and then clinical evaluations, including the complete medical history interviews, comprehensive physical examination, and pure tone auditometry. Then otoscopic examination was performed to exclude the hearing loss due to infections, trauma, and other environmental factors. The vestibular function was evaluated by medical history, inquiry, and behavioral testing. This study reveals that the approved by the Research Ethics Committee of Peking University at China. Then the next, the genetic analysis. In genetic analysis, genomic DNA was extracted from the peripheral blood samples of the enrolled subjects by using the blood minikit which was obtained from Krachen, Shanghai, and targeted next generation sequencing was performed in the proband from each family, and they are including in the whole family the customized capture panel which was targeted the axonic region of 414 non-deafness associated genes in addition to the axonic region. The panel also included all intronic, intergenic, and non-coding regions containing variants that reported in 147 genes and 751 variants, and candidates' pathogenic variants were defined as non-syndromic, including the variants and defined as the supply site and indels, variants with minor allele frequencies. Then the in silico tools mutation tester that was also performed. Then the co-segregation of the deafness phenotype and the pathogenic variants was confirmed in participating the family members by PCR amplification. Then we have also revealed that by the Sanger sequencing, all these supplementary and the pathogenic variants which were related to the nonsense, mid-sense supply sites and the other variants as well. Here is a whole number of different family which was obtained from Okada, and the different patients they are including in this family. Here are the results. In results, we have discussed about the clinical characterization, identification of the pathogenic variants, and identification of second pathogenic variants. We will go in detail one by one. In clinical characterization, there are at least 7 subjects in family which were obtained from Okada, and they were performed as basic functions bilateral, prelingual, and severe to profound the sensorial hearing loss. All affected subjects, which were including in the Okada family, they affected the subjects in the family were born to parents with consequence marriages. No ear malformation, vestibular disinfection, and developmental abnormalities are syndromic symptoms were identified in all enrolled subjects. Next one is the identification of pathogenic variants. The identification of pathogenic variants, they are targeted by the next generation sequencing of 414 non-deafness genes were performed on probands of 3 families. Homozygous variants, which were related with the leucine, tecta, and myosin proteins, and hepatic growth factor, they were identified as the candidate pathogenic variants in the proband of family 4. In family number 1, which was originated from the Okada, all candidates variants having minor allele frequencies lower than 0.0001 in the public database of genomic AD, and the categorized as likely to pathogenic based as guidelines. The third one is identification of second pathogenic variants in family. Okada Sanger sequencing performed and confirmed that homozygous variants which were related to the leucine and surface proteins, they are present in the tecta and the deafness in all participating members in family of Okada which was obtained. Here are some results which were obtained, and here we can see the control and the patient comparison. we can see here the thymine is replaced by the cytosine and cytosine is replaced by the thymine. All these are factors which were elated in the normal person. They are changed by different variants. In another example, here is another change which was arginine and it must be replaced by the guanine. Here is the whole different patients which were obtained from different families, and these variants they including and they are compared with the normal person and control, in which we can see here is the change in the variation. There are the variants which are obtaining from these families, and it causes the basic problem in the deafness. Next, we are going to conclude this presentation. Here is, in conclusion, we can conclude that hearing loss is one of the major disabilities worldwide, which is often induced by the loss of sensory hair and the spiral ganglion neurons in the inner ear of cochlea. Spiral ganglion neurons, which are related with the hearing loss, could be caused by the genetic factors, aging, chronic cochlear infection, infectious diseases, ototoxic drugs, and noise exposure. And genetic factors account for around 70% of the hearing loss. In the present study, we are reporting the genetic cause of the prelingual, severe to profound deafness in the three consequence families through the targeted next generation sequencing approach. And we are also reporting that the consequence marriages that should be comparable, and if there is any endocochlear potential or in the cochlea reduced function, that must be replaced and that must be in domain of our specific individuals. Thank you very much for your attention.